Yeah, so our next speaker is Luis-Miguel Chevin from Montpellier. And just
again, yes. Okay, so hi everyone. So for today's lecture, I'm gonna sort of follow up from where we left it last week, and it also have a kind of nice transition with the previous talk, which was about selection when there's an optimum phenotype, and now we're going to try to get interested in what happens when you have changing environments that are going to move this optimum phenotype. So I think initially, it's worth asking the question, when do organisms need to adapt? We're talking about adaptation. When does it occur? I think it's a legitimate question when you're thinking about these topics. So lots of the things that might be going on may be what Dan Fisher was telling us about, all these kinds of internal feedbacks, where basically some aspect of the evolving population causing a selective pressure on itself. So the population is evolving, and this is causing its own selective pressure. And there are lots of situations that lead to that, such as ecological scenarios, such as resource competition, which causes negative frequency dependence. This has been studied a lot. There can also be mimicry, for instance, warning signals. This is causing positive frequency dependence. For instance, in this case, so this is like this example of Mullerian mimicry, where these butterflies have some warning signals that tells the birds that they're toxic. Now you have to share the load of educating the birds that learn by predating on them, and they learn that they're toxic. They avoid them. So if you can share this load, you have a lower load, actually. This reduces your fitness less. So in this case, actually, the local optimum phenotype is determined by the most common morph. This has been shown that the fitness, so this is predation rate, so this is lower predation means higher fitness. So they had made people actually in where I work in Montpellier had done this experiment where they placed fake models of butterflies, and they showed that if they matched the locally most common morph, then they were less attacked. So really here, so this means evolution of the population will drive its own selection in some way. What's been most common is what's selected. So you have this frequency-dependent type of situation. In other cases where you have this kind of evolution of the population causing its own selective pressure is when you have all kinds of genetic conflicts. This is not what I'm going to be talking about later. It's just that it's a big part of selection too. And so meiotic drive, sexual antagonism, where you have different effects of genes on males and females, and this can lead to some sexual conflicts and other types of genetic conflicts. So this is one thing, but yes?
What do you mean by selective pressure on itself?
Okay. I just mean that the selective population, what you're interested in is what's causing population might be stable, but then it's selected directionally, for instance. The phenotype is selected to change. What's causing that? One reason might be that the change in the population itself, some change in the composition of the population, is changing its selecting pressure that it's experiencing. Let's say here, you change what the mean the most common morph is in the population. This is enough to change selection in the population. You see what I mean?
Contingent on the environment, right?
Also, yes. That's where I want to get. But I mean, ideally, you could think of something that keeps evolving. That's what Daniel was arguing, that could keep evolving without any external forcing, just with this kind of feedbacks. But of course, external forcing, the external environment, is likely to change what phenotype is optimal. If you go back to this example, textbook example that I showed last week, and Enrique showed some of that too. So you have this Darwin's finches where you have different sizes of seeds. And so even within a species, you have like this variation where the smaller beaks are mostly the larger beaks are the only ones who are able to crack these large seeds. They have been tracking the environment very closely in this island. So these are precipitations per year. And then this influences the relative proportion of large and small seeds and translates into different selection on big size. So this would argue that you have kind of the whole story here almost, and especially considering that they also went to the genetics of these traits. Okay? There are many examples like that, but this is one that stands out because it's in natural populations. It's a somewhat close population. The environment has been tracked, and how it relates to the selective pressure is understood. Right? The point is, even when you have internal feedback, such as frequency dependence and etc., it may need some environmental change, environmental variation to maintain the fluctuation. It's difficult for environmental feedbacks for themselves to keep ever, feedback for themselves to provoke long-term evolutions. For instance, this is what we were studying in this in this very simple model that was inspired by the Yeaman walking sticks that Patrick Nozil works on, where they were claiming that it's negative frequency dependence that led to this very drastic fluctuation that alternate from increase to decrease every year very predictably. Actually, with the strength of negative frequency dependence that they had, this would lead to a constant equilibrium. These oscillations would be damped. You need some environmental forcing, even if it's just random noise, to maintain these long-term fluctuations that are actually predictable because of negative frequency dependence. This is just a side story for those interested in that. But this is called stochastic resonance, I think, when you have some noise that reveals the intrinsic oscillatory behavior of the system. So anyway, it's hard to imagine long-term evolution or adaptation without having environmental forcing, environmental change causing it. And so we know that natural systems are characterized by many types of environmental change. You have these periodic cycles, such as this, seasonal patterns of temperatures, which actually also has a trend, as you can see, it's increasing over years, but this is not what I want to show here. And then when you look at like global temperature over years, you can see these trends, that everyone knows about, global warming. There's also quite some noise around these trends. So all these patterns, it's interesting to understand how they affect adaptation of populations. And as we've seen before, a popular way to study that has been with these models where there's a moving optimum. So the fitness function is this Gaussian fitness peak. And then there's this phenotype distribution in black. And then the optimum is moving in time, and the phenotype distribution is tracking this through evolution. So this function is used for mathematical convenience, mostly, but it's also a good approximation. We'll come back to that. And also it makes logical sense. Lots of people, I discuss with lots of empiricists. Sometimes I want to be, just like Nick was saying, we want to be skeptical about this. Of course, we know it's not the truth, that there's a Gaussian fitness peak. It's an approximation. But actually, lots of even empiricists will talk about the optimal phenotype all the time. It's sort of this idea that comes back very often, even if you don't have evidence for it. So what's the evidence for that? I would say there's different lines of evidence that you can put together. The first one is what we were describing. So it's what Nick was describing is this, I would call that direct evidence, is like phenotypic selection analysis. And so this is this Landy-Arnold approach. You're going to miss lots of the references here because of that. That's what it is. So there's this quadratic, sorry? Yeah, censored, I guess. Yeah. Especially, this is very informative to know that ICTS is talking. So the quadratic selection gradient for one trait, it would be just the linear regression slope of the square deviation from the mean with relative fitness, right? This is how it's been defined here. So it's the covariance divided by the variance of something that's squared. So, so usually a negative curvature is negative curvature, could be like that, could be like this. It's interpreted as stabilizing selection, whereas a positive one is interpreted as oh, yeah. Top corner would be the best, I think, if you can. If I remember well, I don't know. Top. Yeah, top right. Thanks. Thank you. So, thanks. So actually, this meta-analysis by Kingsolver et al. In 2001, and there's been some others after that, because many studies have measured these effects. They didn't find the prevalence of negative ones. They found there were as many negative as positive. So this could mean that there's lots of disruptive selection, and as much as stabilizing selection. Yes? Yeah, I agree. But I've been wondering, how do you show that there's a peak? It has to be, you have to be on both sides of the peak. Yeah. So I think I'm going to try to go towards that, but let's see if this.
So one potential explanation is that if you have multiple traits, right, people usually read the lambda off the diagonal of that quadratic matrix, when in fact, what you probably want to do is look at the eigenvalues of that. So that may give you a large one, when you look at the eigenvalues, a large negative, and then a bunch of tiny positive ones, which are probably noise.
So I was sort of trying to argue that gamma is not great for measuring what we want, but because for for the mean phenotype, the delta z bar equals VA times derivative of log fitness relative to the mean phenotype, which can write as VA times 1 over W bar d W bar over Z bar. This is beta, okay? This is the selection gradient that comes in the theory. Just happens to be exactly covariance of Z with W over W bar, right? This you can show. Did I say something? No, divided by phenotypic variance. Someone correct me if I'm wrong. So it's a regression slope, covariance divided by variance, a regression slope. You can show that for normally distributed trait, if you really go, I mean, there are different ways to show it, basically. If you write mean fitness as an integral of the phenotype and you differentiate the normal distribution, et cetera, you get this. So it just happens that the change in the variance is not, this is not true for the change in the variance. When you do the covariance of Z minus Z bar squared with W over W bar, divided by the squared variance, etc., it's not what predicts the change in variance. So it doesn't have this elegant thing that beta, which made beta popular, I think, which is it directly connects to the theory. This doesn't. This is just to me, this is just convenient because you take the square deviation from the mean, you put it in your regression, and it's easy to get. But it has, then the change in variance will have this thing. So this is like divided over sigma to the 4. I guess this is gamma, like that. But the change in variance will have, I'm writing VG, okay. Change in genetic variance or phenotypic variance. Both actually, I think. What is it? It will depend on gamma minus beta squared. Times VG squared, I think. Something like that. Sorry. You remember, Enrique, you had that on your... It looks something like this. Well, my point is that it has the beta squared in it. So, and every prediction you will have from theory will actually... Okay. I'm not going to do this on the blackboard because I'm not good at that. So, and so in Landy-Arnold, they had some argument for why there's this and stuff, but I think it's just a convenient way to estimate things. I have something on my next slide that sort of... So basically the...
By the way, the easy way to think about that is if gamma is 0, you're still going to lose varying because of directional selection.
Yeah, but I don't think it's because of directional selections. That's the point. You're losing variance because the derivative of log fitness relative to, I mean, it all makes sense if you don't use gamma, actually. It's not direction. What you're saying is that you would lose variance with a linear fitness function, which has this term, with a linear fitness function, this disappears, and all that's left is that. But a linear fitness function is not linear on the log scale, so actually it makes sense that it reduces the variance, because an additive increment is not the same when your fitness is 1 than when your fitness is 10. So only the exponential fitness function is something that does the same proportional increment. Oh, great, thank you. So, yeah, I was, but then it got stuck. Yeah, but it got stuck. That's what I was doing, thanks. So with the Gaussian fitness peak, as we showed just earlier, as we discussed earlier, actually you have gamma positive when the mean phenotype, gamma changes with the deviation from the optimum. And when the mean phenotype is sufficiently away from the optimum, then gamma becomes positive. And so actually, when for instance, I took this, this paper that deals on laying dates in gray teeth, sorry. It was a science paper about this plasticity in laying date, etc. And they had, in different years, they had this linear, different, sorry, I don't have the axis here. I think it's the laying date. They know what should be the optimal laying date, but it's the laying date of the birds, right? And this is how the linear selection gradient depend on that. And this is how the quadratic gradient is changing with that, right? And if I take this and I assume that this is all caused by a Gaussian fitness peak that's moving, and so this is when you're away from the optimum, and this is when you're at the optimum, I just did it by hand, I just took the slope here, and this is what I get for the quadratic gradient. So I haven't plotted this on here, but it actually seems to fit the data even more than their line, So to me, this is all consistent with the Gaussian fitness peak that's moving. Gaussian fitness peak is not great, but it's always better than a quadratic thing, because this can become negative. So negative number of offspring, at least this one is like that. The quadratic one is good for your log fitness, for your relative growth rate, like dn over dt. Let's say dn over dt equals Rn plus things. This thing is a relative growth rate. This can be positive or negative. Here you can have this quadratic function. It's equivalent to the Gaussian for the number of offspring. You see? I don't know if everyone's following me. It's called Malthusian fitness versus multiplicative fitness. Anyway, so to me this is, and I think it seems to be consistent with that. And so more generally, what appears in theoretical predictions is always this theoretical predictions for the variance we've seen just before, but the ones I'm going to talk about are a lot about changes in the mean phenotype. They're all in terms of the strength of stabilizing selection, 1 over Vs, the width of the fitness peak, which has been introduced before. I've never seen many predictions based on this thing, gamma, which happens to be, so beta square minus S. So you can write, what I did in the previous graph, it depends on deviation from the optimum, or the mean phenotype from the optimum. So why not estimate that directly instead? This is what, just like beta was what appears in prediction for the mean phenotype, 1 over Vs is what appears in prediction, it's not gamma. So this actually comes out naturally when you're using GLMs. So this is what people are actually doing. People working on quantgen in natural populations, they measure a number of offspring, for instance, on this laying date. The number of offspring is Poisson distributed, and then you use the log of the expected number. So this is Poisson regression is kind of a standard thing, actually. And if you do a quadratic model on this log scale, then it's actually a Gaussian fitness function. It is. So this comes directly. You can just retransform your slope and quadratic parameter. The quadratic parameter will be the same. You have the strength of stabilizing selection directly as in the models. And then you can use the slope to estimate the optimum. So we have done that. And then alternatively, you can actually fit the actual Gaussian fitness peak with the optimum, in terms of the optimum, the width of the fitness peak, etc. It's no longer a linear model because you have products of the parameters that come in, but you can estimate this in frameworks such as STAN or these types of framework that accommodate any types of function. So, so we've been doing that. with respect to flux, okay. So if we go back to this idea, there's this Gaussian fitness peak is better directly estimated, and then it might be that lots of what we'll see as variation, you can interpret this as just constant width and something that's moving. So then if you're having this framework, you had much more power to estimate this width of the fitness peak and strength of stabilizing selection. And this might be why here, I mean, in any single estimate of gamma could be positive, negative, changing across years, and then you just have 1 year. What if you put all years together and you assume that it's the same width of the fitness peak and it's fluctuating, just like in theory? And so this is what we did here. We had many long-term data sets of birds and mammals in the wild. So there's this whole group of people working on, so there's the soy sheep, bighorn sheep, great tits in the Netherlands, Oxford, all these big data sets that have been running, so from 9 to 63 years, so the average was 33 years. And so we just estimated this with the idea that, so this is the red is the fitness peak, and the green is the phenotype distribution. So the gradient that you would get, you know, the linear gradient, depends not just on fluctuation on the fitness surface, but also on changes in the phenotype distribution. In this case, they were affected by plasticity, which I'll talk about on Friday. So the selection results from changes in the phenotype distribution and changes in this fitness function. And so we ran several models with or without fluctuation, with or without an optimum, etc., or with the quadratic term, the log scale. And we ran that across all these data sets, and then we computed a score for how well supported the models were. It was based on an equivalent of Akaike weight, but a bit more refined than that across data sets. And so we found overall that models with an optimum were more supported than models without. I mean, this is across birds, mammals, and in total. So on my weights were just per study. So the flat priors, you mean across models for different models?
Yes, in different models. There's no way to consider all.
It was based on the. It was just something based on the exponential of the maximum. Sorry. The minimum AIC or something, but it was not AIC that we use. It was like something that's cross-validation. Pierre is the one who would know more. I mean, I followed when we did it, but now we'll be able to explain the whole detail. Leave one out, validation criterion, and then we use, I mean, I remember we did some kind of equivalent of Akaike weight with the exponential of that, and we did it carefully at the time. I sort of proposed the thing, but then I wouldn't be able to explain it to you again. It was a way to correct for the number of different number of size of different data sets. Yes? Yeah, okay, so this was constant. This was fluctuating, not autocorrelated. This was fluctuating, autocorrelated. I don't want to go into the details because I explain some of that later. All my point is that models with an optimum were, well, they were at least majority, but they weren't very drastically more supported. But let's say the light red is the equivalent of the light blue, but without correlation. But in the ones where the model with optimum were the preferred one, then we could estimate the mean optimum, which was earlier than the actual laying date, but not significantly in mammals, but significantly in birds, which consistent with what we know that there's lots of these birds have directional selection for earlier laying date. Then we could get the variance or coefficient of variation of the optimum, which wasn't very large in this case, so it wasn't much larger than the mean deviation. So this means that fluctuation in the optimum did not really cause changes in sign in the direction of selection because it didn't fluctuate both sides of the optimum. And this gets a bit into what Mike was saying, that how well can you tell that there's an optimum in any year? Actually, you're like this or like this. It's rare that you have these really things on both sides. And we were arguing about that with Losco Crook and other people. Some data sets, you clearly have things like that. Others, you sort of can say that there's an optimum, but using all years together. So anyway, and then we got the width of the peak. So if we square that, 25, this is about what VS would be this squared. So it's like similar to what what people are using actually. Okay, so this was just evidence number one. I'm getting super late. Okay. Evidence number two. I just stole this reference from what Mike showed because I was interested by this paper. So this is like these cellular traits. I mean, there are other cases like that. This evidence I'm talking about is comparing mutational to standing variation. How much variation is produced by new mutation, and how does this compare to what's in natural populations? And so they measured many traits, cellular traits in this spindle during early development of this Caenorhabditis. And so they also had these mutation accumulation lines here. So you see that variance at generation 250 was larger than initially, so they could estimate how the variance increased for many of these traits. But then what they did is like, they couldn't explain basically the standing variance. They could only explain it from the mutational variance if they included stabilizing selection. And actually correlational selection, which we hadn't heard. I thought you would have heard about that when I give this lecture, but correlational selection is just selection for combinations of traits that don't deviate much from the optimum or something like this. So that's it. Standing variation of all traits on the y-axis is well predicted by their mutational covariance on the x-axis, but only after account for, so they had this stabilizing selection, correlation selection matrix. So there are different flavors of that to compare variance, standing variance to mutational variance. So I call that semi-direct evidence because it's really kind of very closely connected to the prediction of the model. Another evidence comes from distribution of fitness effects. So I mentioned this last week. If you have a Gaussian fitness peak and you look at selection coefficients as differences in log fitness between two genotypes, one of which has so there's mutation effect on the trait. So sorry for those who weren't there, but you have to catch up. So if you're at the optimum, then there are only deleterious mutations. But when you're away from the optimum, there's this fitness distance S0, which depends on how far you're from the optimum and the width of the fitness peak. And then we'll have some beneficial mutations up to S0, which bring you back towards the optimum. And then still deleterious ones. So you have beneficial and deleterious. The whole shape should be a reverse displaced gamma distribution, which comes from a chi-square, basically. It's really a square deviation from the optimum that leads to that. And so this predicts that under increased stress, you should have larger variance of selection coefficients, higher proportion of beneficials, keep the same mean, okay? Well, people have tested that, some of that directly, like this experiment in Isabel Gordo's group. They had effects of mutations, single mutants that they had either in control medium or in a minimal medium with fewer resources, or that plus high temperature. So the stress is increasing this way. And you can see that the proportion of beneficial mutations is increasing, and this distribution is spreading. So actually, they explicitly were testing these models. So they estimated the parameters from distribution of fitness effect, this shape of this gamma distribution, etc. Several of these predictions made sense. Yes?
Prediction independent of your assumptions about the distribution of incoming effects on the phenotype?
It assumes a normal distribution of effects on traits, which if you're willing to believe Guillaume Martin, he had a model that he had a model where this sort of comes out from many regulatory and developmental integration of underlying traits, that the traits are actually under stabilizing selection, are made up of many other traits that get integrated, and through some kind of central limit thing, it leads to a normal distribution. You can read that. It's in 2014 in Genetics. I mean, some people got convinced by it, so I think there are some convincing arguments to that. I don't know if it's, I mean, yeah, it's always the question of how robust is it exactly Gaussian here, the shape? Is the distribution of mutation effects, does it have to be Gaussian? What if it's just symmetric but not Gaussian? How does this change? I don't think it would change qualitatively that much. What happens that you have more beneficials when you're away from the optimum? Yeah.
Maybe I missed this in your answer you were giving. I don't understand the excessive beneficial mutations that you have when you move away from the optimum, because I thought it was symmetric exactly, like the deleterious mutations would be deleterious because they either decreased or increased the trait, right?
Here, look, the blue ones are beneficial. If you have this...
But they could be on the other side of the mountain, so if you...
So if this is the population, the wild type, if it were at the optimum, you can only go down.
Okay.
Mutations can only go down. If you're here, some of these blue mutations, they go up, and the red ones, they go down. It's just this. It's quantifying this. So these guys in Jeff Jensen's group in Lausanne at the time, they did the same with a different approach of measuring. They had this bulk measurement where you put all genotypes together. So they came up with a nice way to measure fitness of many genotypes. And so it's the same, basically. A standard, elevated temperature, elevated salinity, both. So this is increasing stress. And so they sort of estimated also the S0, the maladaptation that would be consistent with that. So they also, this you can't read, but this is the proportion of beneficial ones is increasing, et cetera, et cetera. I don't go into detail, it's just to give you the... So another one is the paradox of stasis, which was what Nick Barton was referring to. If traits are not evolving over the long run, it must be that they're under stabilizing selection. So this is like a kind of verbal argument that has been made more quantitative and explicit in a series of papers, especially these ones, where they actually used models from quantitative genetics, simple ones that assume constant variance, etc., and to say that this rate of evolutionary change. Okay, so these are patterns of evolutionary change over the time interval over which they're measured. So this compiles, field studies, fossil time series, and then phylogenetic divergence, everything on the same. And so you see that you can, even though you can have somewhat a rapid change over the short term, it doesn't accumulate, doesn't increase when you... So I think Mike presented similar types of things, but this is pulling many, many data sets. And they tried different models of adaptation to an optimum, and they said that this is consistent with stationary fluctuations of an optimum phenotype, plus some rare shift that occur every million years or so, then you have this long divergence. But other than that, it's consistent more with fluctuations of an optimum than with drift, for sure. Drift would have a variance that increases linearly with time. I'm sorry. Divergence. Sorry? What is paradoxical? The paradox of stasis? Oh, the paradox is because you have lots of response to selection in the short run, as we've been discussing for the week, but then you don't see phenotypes diverging among species over the long run. Based on that, you would expect that these explode. So it could be either that they're mutational limited, it could be that they're stabilizing selection. I mean, it's still debate. There was a paper on that like yesterday in Science, I think. But I haven't had time to check it. So, okay. Another line of evidence comes from so Nick was saying when you select something, it usually stays there. Well, not when it's a very, very conserved trait. So you look at these traits, like this is like allometric relationship in wing size in Drosophila. And these are, so allometric relationship is the relationship between the log of one trait and the log of the other trait. And so this is log of, I can tell, well, these different traits, you see, this different parameter of wing shape. And so they're very strongly correlated. So this is the people who work on that, they study that within species, cross species, etc. You have ontogenic, this shows up in ontogeny, shows up in standing variation, shows up across species. And so they had 111 species, and this is the slope that each line is within species variation. So they're all along these lines. And then across species, they're also along these lines. It's super conserved. Developmental constraints. I mean, this describes, this shouts developmental constraint, you cannot escape from it. Well, they decided to change this slope. So it's a selection on something's covariance, right? So they did correlational selection, it's called, but they use disruptive, this is for people like that, they use disruptive correlational selection to not deplete the variance. They had this saddle fitness function, so they selected the one that, so this is the, what is it? Sorry. So the dashed line is the allometric slope, and then they selected for this other line. The individuals that were closer to these lines, they kept, so the one in black. And they also did down selection, you know, have a flatter slope, okay? So this is the same group that has this paper that I told you yesterday. It's people in Norway, mainly Tom Hansen, etc. Christophe Pelabon. And so they got lots of responses when they did it. They got a selection response on the intercept here of this curve, and then on the slope also. So high and low. So there are males and females, right? But they're selecting high versus low on the intercept, high versus low on the slope. They got a selection. But then when they relaxed selection, they saw it coming back, especially the slope, quite rapidly towards where it was. So this could be due to the breakup of LD, etc. They were trying to be careful about this during their selection. They come up with some numbers that here it could only account for at most 20% of this response, just the fact that you build up LD through selection and then it gets broken up. So I guess the idea here is that this trait is not that it's constrained developmentally, or it's under very strong stabilizing selection. I guess the difference is a bit subtle, you know. If to select on that, you need to perturb the development so much that if you relax selection on it, then it will sort of come back to where it was before. Okay, 6, and then I'll stop because I have other things to say actually. This one is like ecological speciation. I think also has something to say about that. So people study these different ecotypes, and then that specialize on different types of resources. And but how do you know if they're fit? You know, because there's not that much variance. When it's forming species, you can still cross them. Some ongoing ecological speciation. So this group here, they did that on adaptive radiation in pupfishes. And so what they did, I thought was clever. They did this recombinant F2, and when you do the F2s, you generate lots of variance because you know you've seen that before. So you have some lines that have lots of pluses and minuses, and others have pluses and minuses. Other loci, you end up with some genotypes when you recombine them, they get all the minus and all the plus. And so you have all this variation, the segregation variance. So it allows you, then you have all these different phenotypes, it allows you to measure the fitness landscape. And then when you place the original ones on this, they sit on at least this one and here, they sit close to this fitness peak. So it means they were actually at their fitness peak. Okay, so this is like, you rarely have all these lines of evidence in the same study, that's the problem. I would argue with this, when you combine all this plus others actually, which is like things that are under artificial selection, like salmon, they're in lakes and sometimes they escape. They impose a strong load on local populations. So it's sort of, on wild populations, also suggests that they're being pulled away from their optimum. So this, I would say that stabilizing selection seems overall well supported. So I hope you didn't mind that I went through all these things, but I wanted also for myself to try to think about what we have for evidence for that. Of course, the specific shape may deviate from what's usually assumed, especially when you're far from the optimum, but yeah. I would say that it's more than just it exists, but paradox of stasis, I mean, it's traits that have been chosen randomly, and they behave like that. Yeah, that's what I would think, yeah. People who measure the distribution of fitness effects across environment, they had no traits in mind, you know. So if you can come up with another model that does these fine predictions of how fitness effects change with the environment, I'm willing to use it, but okay. Yeah, that's the thing. People don't mean the same thing with direction with stabilized selection. I just mean that there might be some optimum in your displacement. I mean, if you're stuck here, then you're saying, okay, there's not much evidence. I would argue that even this first thing I don't really trust because of that, and then the rest. I mean, it doesn't apply to any trait, etc., like the mutation rates. No, you have to be skeptical. I wrote this because I am skeptical, you know. I'm using these models all the time, so I want to know if it makes sense. Anyway, so here the goal, I'll try to go through how can these moving optimum models help predict adaptation to a changing environment. I'm pretty sure I won't be able to finish now, and I'll go through these different types of environmental change, okay? So, but the good thing is that this has been introduced before, so I can go rapidly. Just to say that when you have an optimum, actually, when you do a Taylor series on the log scale for something that has to be positive, it's kind of more meaningful. If you see what I mean, fitness, if it cannot be below 0, then you do the Taylor series on the log scale and you put it to the second order, then it produces that. And so this is kind of it can approximate Gaussian fitness peak with an optimum. It's better than the quadratic approximation when you're far from the optimum. It will have to be. If the trait is normally distributed, because it's polygenic and has residual variation, then you integrate the product of the Gaussian phenotype distribution and the fitness peak. This is also Gaussian, as we heard before, I think. Now it depends on the deviation of the mean phenotype from the optimum. And this omega here gets replaced by this VS, which is, this should be, I think that's it. No, that's right. So it's square root of width of the fitness peak plus phenotypic variance. So when there's more phenotypic variance, it kind of flattens the fitness landscape. As per, yeah. I was just saying that variation in directional selection that you see, it switches from there to there, it may be because the optimum has changed or because the phenotype has changed. In our case, in lots of cases, I would argue on Friday, you have phenotypic plasticity. When the environment changes, the mean phenotype is going one way or the other from the optimum. This might be what's causing change in selection, even if the peak hasn't changed. So if you have estimated the fitness peak and the change in the mean phenotype, you're better off than just estimating the local slope. Yeah, that's the argument. But here it wasn't the main point I was making. It's just the graph had that, so I copied the graph. Okay, so then the mean mismatch with optimum, which I write as x here, z bar minus theta. This is what drives the dynamics in this, or the mean phenotype. If you think of a mutation that has an effect alpha on the trait, and you're in a haploid population, then I like using this thing for changes in the effect of selection. It's been used by Fisher and other people. It's kind of convenient because you can show that the change in p over 1 minus p it just depends on the log ratios of their fitness. In a haploid, for instance, in a haploid population, p prime equals p w, how do you call that? A over W bar, and Q prime, which is 1 minus P prime, equals Q WA, or let's put it the reverse, over W bar. And then when you do P prime over Q prime, it's just PQ, P over Q times WA over W bar. W. You cancel out the mean fitness. Then you take the log of that, it becomes log this plus log that. Log that is log of this ratio of fitness for lots of things. For instance, in selective sweep theory, that's what's used a lot. It's just a more convenient way to measure selection gradient. Just happens that in this case, with this Gaussian fitness peak, it becomes just this. And so it depends on the strength of stabilizing selection and the squared effect of the mutation time, this term, which is the product of mutation effect and deviation from the optimum. But for us, what's interesting is that it's linear in the mismatch with optimum. So it depends additively on that. And for a normally distributed trait, the selection gradient on the mean phenotype, which is this derivative of log mean fitness relative to the mean phenotype, it's also just minus S times X. So it's all linear in mismatch, so it makes simple models that we can analyze. That's why we like it, actually. But then we try to see that it works empirically. But then the response to selection, it's product of the genetic. So I'll just write G for the additive genetic variance. G is VA. Because when you have multiple traits, it becomes a G matrix. It's just one letter, it's simple to write. And so G beta is just minus GSX. So it depends on deviation from the optimum. And it's larger if there's more SG, which is something like the adaptive potential. The strength of selection times how many variants there is to respond, you know. So if there's narrow fitness peak and large additive genetic variance, it responds faster. So and in cases where genetic variance can be approximated as constant, then you have a very simple dynamical system because it's a linear differential equation, etc. And there are lots of things you can study. So you can make analytical progress. So you can use the hypersimplified model for genetic variation, but then plug it into more interesting scenarios of environmental change, for instance. Okay? And then you can try to validate this with simulations, for instance. All right. So any questions about this? Is it fine? Okay, so I'll start with the directional environmental change. So you could separate directional into abrupt change, like an environmental shift. This has been somewhat addressed in previous lectures. I would hope it would have been addressed more, but I talked about this in my previous lecture a little bit. And you have either the adaptive walk regime of R, or you have this polygenic regime that we discussed in reference to Laura's paper also. I'll come back a little bit to that, but I want to move on to like more gradual tendencies such as global warming. They're usually modeled as a steady change at constant speed, so you have this linear increase in time that follows something where it was more or less constant. So you can see here that how this could work as an approximation. So first, in the kind of low mutation regime where you have these adaptive walks and successive substitutions of mutations, if you remember, when you have a sudden shift, R showed that initially you can have large steps that are taken because you start far from the optimum, right? And so this is what, and I think Sam showed that again yesterday, so you've seen that several times. This is like the spacing between steps, so the biggest steps can be large. It doesn't have to be exactly the first one. What happens now when you have a gradually moving optimum? Well, it's not the same because now the selection coefficient change as the optimum is moving. And a mutation first needs to become beneficial. So if you think of like here, the background phenotype is at the optimum, it's this green line. And this black mutation here, it's causing you to have this fitness, right? And so this is the effect, the fitness effect of this mutation, okay? Initially, it's deleterious. Now the optimum is moving. It's still deleterious. But so you see this is the fitness of the wild type, and this is the mutant. So as time approaches, this deleterious effect decreases. And at one point, you will have higher fitness than the mean phenotype. And so it's becoming beneficial. So there's this waiting time. This waiting time will be smaller if the mutation has a lower effect. And so this is what Joachim and Michael Kopp and Joachim Hermisson studied in a series of papers. And they show that there are these 3 different time scales where first this mutation needs to become beneficial. So this is the changing selection coefficient of the mutation, as I explained. And when it hits 0 here, this is the first time, the waiting time until it becomes beneficial. Then the mutation is beneficial, but it's not spreading. It occurs repeatedly and needs to escape drift, as we saw last week. There's the biggest probability is just that it's lost when you So there's this waiting time until it escapes drift, which happens here, and then it starts sweeping. And then there's the third time is the sweeping time, which here they measure as the time to reach half frequency. So it's kind of half of the total sweeping time. So there are some orders of magnitudes for that. And so based on this, they show that slower environmental change will be dominated by this waiting time before you become beneficial. And this favors small steps because they become beneficial earlier. When you don't move away from the optimum, then a lot from the optimum, then it's, well, based on my drawing from before, for my cartoon, they become beneficial earlier. So here they simulated, for instance, slow versus fast change, and you see that under slow change, you have these small steps that are taken, whereas under fast change, you can have this faster change. And so overall, this boils down to a single composite parameter that determines the genetic of adaptation in this context. And so it depends on, well, you could argue this green thing is the ecological part, is the speed of environmental change, whereas this yellow part is everything that has to do with the provision of mutations. So number of mutations that enter the population depends on the population size times the mutation rate. Then there's the strength of stabilizing selection, and then the effect, the standard deviation of the effects of mutation on the trait. Was it discrete time model? Yes, if it's a discrete time model, then everything is in generation times. If not, you have to specify. So let's say yes. But it's not very, it doesn't have to be. I mean, you can, as long as everything is the same units. If this is not in generation time, then even the strength of selection will not be in generation time, you know? You can measure strength of selection in per hour change in the trait. There can be a time component in continuous time models. Everything has a time component, actually. Mutation rate per unit time. You see what I mean? So this is why lots of people prefer using discrete time models, because they don't have, it's all in generation, units of generation. So with large gamma, the environment changed fast relative to the adaptive potential, and then adaptation is genetically limited. This is what Orr was studying in the limit of very high rapid change. Then it's really, you need to wait for the next mutation to occur. It's not that you need to wait for the environment to make your mutation beneficial. Small gamma is a reverse. The adaptation is environmentally limited, and mutation of small effects fixed first, right? Okay, so this was in this adaptive walk regime. Can I switch? Yeah. Okay, now in this more polymorphic regime, polygenic regime that I was describing before, what happens in this case is that the distance to the optimum initially increases as the population, the optimum is moving. But then when the distance to the optimum increases, this increases the strength of selection towards this optimum, because the deviation from the optimum is increasing. And so this increases the selection response. And so you can plot everything in terms of this deviation from the optimum instead of the actual trait. And so what you see is this. Basically, you start at the optimum, and then the distance to the optimum increases, but this increases the strength of selection. At one point, it equilibrates, where at a place where the change in the mean phenotype is actually equal to the speed of the environmental change. And so when you solve for that, you get that the equilibrium distance to the optimum depends on, so it's negative because it's lagging behind, and it depends on the speed of environmental change and the adaptive potential. And so this is, so yeah, I had this thing where I explained where this is coming from, but I mean, you can write the whole recursion for that. Perhaps I can put that in the Slack in the end. The recursion is quite easy. So this curve is like the analytical prediction for what it does. And one kind of fun note is that it's kind of the exact reverse dynamics than the sudden shift. The sudden shift, you start away from the optimum, and then you approach it with an exponentially declining distance. Here you start at the optimum, and then you get away from it until you reach an equilibrium exactly with the same dynamics. It's the exact opposite. Okay. I want to highlight the parallel between this thing. I just use this color code like environmental change, adaptive potential, what we had before in the other regime, environmental change, adaptive potential. And actually, it also, when this gamma parameter was larger, you also had a larger lag here. So it sort of says the same. It's consistent across regimes. It's just quantitatively, it's not the exact. This is more noisy because you have discrete steps, but it's qualitatively similar. Okay? Yes? Yeah. Yeah, I mean, this is, if you start here, then the population evolves towards the optimum. And as it evolves, the strength of selection decreases, and this is why it's slowing down the approach. And this has been studied in numerous models. And the true dynamics will have some noise around this and things, and some people like Laura and others have modeled this more explicitly, but this is what it is. Okay, so, and then the original models of that were really, by Mike actually and others, they were really interested into what this causes for the lag and for extinction risk, because so this distance from the optimum increases, and then at equilibrium, your main phenotype might be in the zone where you're so mal adapted that the population cannot grow anymore. And so this lag load is just depend on, if you use log fitness, it depends on the squared deviation from the optimum. And so it's just this squared, just this term. From this, you can compute a critical rate of environmental change that population can tolerate without going extinct, which is when the growth rate becomes 0. When the lag is so large, the load is so large that the population growth rate is 0. And what's interesting about this is that this critical rate of environmental change is faster when you have more genetic variance. Okay, if you have more genetic variance, you can adapt to faster environmental change. What's perhaps counterintuitive is that it's also faster when you have stronger stabilizing selection. Why it's a bit counterintuitive is that when you have, so when this is more narrow, a given deviation from the optimum gets you lower, you know, than here, right? So this reduces, for a given deviation from the optimum, you have a higher load actually. But the strength of selection, stabilizing selection also acts on changing the mean phenotype and reducing this lag actually. And this effect is what dominates in here. This is why it's stronger selection leads to allow the population to tolerate faster environmental change. And this happens here actually. You have S here, and you had an S in the denominator that gets squared. And so you still keep an S here. Okay, so I think I don't know if I have much time to describe the next slide, but this, of course, the fact that you have a square root, etc., this all depends on assuming this Gaussian fitness peak, right? If you can imagine other fitness functions where things get more complex. And this is what these people have done here, especially if you consider that the intrinsic rate of increase depends on fecundity, birth minus death, basically. And the birth cannot be negative, so it has this Gaussian component. So the log fitness, if you want, has some Gaussian in it. And so basically in this context, you can imagine fitness function where when you increase the deviation from the optimum, you don't increase selection proportionally. So this is what's happening here. You have this, in the model I was studying, the farther you are from the optimum, the stronger the selection. This is a selection to bring you back towards the optimum. But there are scenarios where there's a maximum for the selection, and then when you increase the deviation from the optimum, selection decreases actually. So this is an evolutionary tipping point, because if you start, at one point the environment accelerates, and then you enter in this regime, then this deviation from the optimum keeps ever increasing, even if it slows down afterwards. So well, this is kind of an interesting situation where you have this kind of extinction vortex, and these things that would be called hysteresis, which is you how could I show that? I guess when you start here, the population starts being away from this point, and then even if the speed of environmental change decreases, it's still stuck in here, and it keeps going farther and farther and farther away. Anyway, this is just to point out caution about an optimum doesn't necessarily mean this Gaussian fit. Okay, another type of environment that's really relevant is this cycling environments. So you can think of cycling, what should it matter for selection? If you're a short-lived organism like a Drosophila, seasonal cycles, they might take 10, 20 generations, I don't know. So you can evolve over this time. These organisms also have usually large population size, so they have high adaptive potential, these small, short-lived organisms. You have other cycles that occur over larger periods, so they could be tracked by long-lived organisms. So, and one example of these seasonal cycles are these chromosomal inversions that Dobzhansky studied in the old days, based on, I think, poly-10 chromosomes. Is that something like you could see them on the microscope, I think? Okay, I have a nod from a Drosophilist, so that must be true. And then they were tracking this frequency change over the years. Recently, this has been done with the genomic approach. And so they tracked so they found nearly 2,000 SNPs throughout the genome that showed this, repeated frequency change up and down between spring and fall in this Dmitry Petrov's group and Paul Schmidt. They argue that it's not. So I remember you said that, and then I went through the paper. They said lots of them are not in inversions, and that's the whole point. Otherwise, it would just recapitulate that. So if it's just that, I don't think it's, but I think they have more than this. You're going to dislike the next slide much more. Don't you? We're on video, so. So later in this system, I think they did this thing that was, first they had this super nice drawing in the paper, and so they wanted to see where this is coming from, so they put flies in cages in the field, several replicate flies over from summer to autumn, I think. And they were tracking what? They were tracking several traits, and they also genotyped them at these different points. Okay, they were tracking. They had replicate cages, etc. So what's nice is that they had traits and they had a genetic change, and so it's hard to read, but let's say that there was some fecundity, there were some egg size, and other traits. And so the different gray lines are the different replicates, and the orange ones are the average among replicates. So it's kind of repeatedly doing phenotypic change across all these traits. And then when they look at genomic change, when they did FST across different times, you can do FST. It's like genetic structure, but across time, you consider different time points as different populations. You say, are they differentiated? There were different. So this is baseline versus last, baseline versus 0.4, etc. So there's increasing differentiation when you increase the time difference, and it's more than in their technical replicates, etc. And so this was, I used this thing to say, this is what they say. So we could debate that, but not during my talk tomorrow about this is. But they say that overall the results show strong and temporally variable natural selection that consistently drive rapid and polygenic adaptation of multiple fitness-associated phenotypes on the same time as scale as environmental change. So this is kind of nice, I think. So these types of scenario you can also model easily by thinking that the optimum is following this sine wave, which has period t, and then amplitude a. And again, if you have a polygenic trait with constant variance, so there's always this transient influence of initial conditions that you want to get rid of because you don't know what the initial condition was. But over some time, that depends on over a time that's shorter if you have higher adaptive potential, basically. The mean phenotype, it will settle into a sine wave that have the same period as the optimum. but it has a lower amplitude, which is given by this. And it's also has a shifted phase. It's delayed, basically. And so here, for instance, you see that when this SGT term is large, then this dominates here, and this tends to 1. So this means that when you have higher adaptive potential and slower oscillation, which means larger T, then the mean phenotype is able to more closely track the optimum, because the amplitude is, when zeta tends to 1, then it has the similar amplitude to the optimum. And the phase lag tends to 0. So this is what I showed here. With a period of 100, you see these waves of the mean phenotype that's a little bit delayed from those of the optimum. They manage to get quite close to the amplitude of the optimum. Whereas when it's faster, they have this lower amplitude, and also they're more shifted to the right, so they're more delayed. Okay. You can also do that with for single locus. If you have a fluctuating selection, cycling selection coefficient, then it's kind of easy to solve too. In this case, I mean, it's if it stays close to half, then it's also a sine wave. If it gets to very high frequency, then it's a bit more complicated. You need to account for anyway.
One interesting thing about these cycles is twice in a cycle, you're exactly at the opposite.
By chance.
Wrong direction.
Yeah. Yeah, it's true. Yeah, for this one, for the previous one, they're well adapted somehow, but they're still lagging overall. So you can compute the overall load that you get when you integrate over a period. And this has been done also by McIntosh and others. When you have a fluctuating selection coefficient, then actually what you get is that the amplitude is also larger when you have a larger period. You can also find this analytically in this case. And the phase is always 1/4. So the maximum frequency is when the selection coefficient equals 0, right? And this is when the selection coefficient equals 0, this is when the frequency starts to decline again. So it's always have this phase shift. And then if you change the period here, you have a slower fluctuation, slow cycle. You see that you reach a higher amplitude. But again, if you reduce the amplitude of the selection coefficient, then it decreases again. So this could be, I guess, compared to what they had in their model. I didn't do it. This I just put together now, but this is for single locus, considering it's not affected by the others. Could also have selection coming from deviation from the optimum. Okay, so the last thing I want to tell you about is randomly fluctuating environments. This is what I've been working on a lot, actually, also doing experiments and things, but I think I won't have time to really go into that in detail. Or perhaps fortunately for you, I don't know. But I mean, even the idea that most environments, when you remove this trend, there's something that remains, right? So you can argue forever, is this really stochastic? Is it really chance? Probably it's deterministic, but if you don't know what's causing it is not in your system. It's like meteorological things you're not modeling.
I guess the key is you have no autocorrelation. The other two models have autocorrelation.
I will have.
They break, you allow
This is all about autocorrelation. So there is autocorrelation, but basically, if you don't know what's causing it and you don't have the precision, effectively it's random for us. We treat it as a random thing. And actually, you could argue that for organisms in the wild, it's the same. It's random. They don't know what's causing it. So it's stochastic process you can describe as it's the extension of random variables over time. When you have series of random variables and you think of how they connect to each other, then it's a stochastic process. And so randomness matters when you're dealing with predictions. Like if you had the deterministic time series and you know that it's going like that, then you can project it. It's fine. You just use this is prediction. The future is certain if you have enough precision, etc., perfect knowledge of all the factors. When it's stochastic, you have measured this. There's no way you can avoid, modeling all these different possibilities. So you can predict things, but probabilistically. So it means that you won't be just interested in the average of the process, but also sometimes in the variance that this will generate across replicates. This envelope here, sometimes you have to account for it, especially for things like extinction risk. Well, drift does that a lot too. When you have drifting thing, that start at frequency of half. The expected frequency is half, right, in the long term. But there will be fixation on both sides, so you need to account for that. Okay, so when you have these randomly fluctuating environments, there's this temporal autocorrelation. The simplest way to define it is, for instance, over time scale, you consider, this is time series that we actually use in our experiments. So the environment at time t plus 1 against the environment at time t, if there's zero correlation, there's no autocorrelation. If it's, this is when it's strongly autocorrelated, it's correlated with the you could correlate it across more time steps if you want. And in fact, when it is correlated over one time step, it will have to be correlated over the next ones in lots of cases. And so this description also relates to how it looks like when you look at them, This time series, when it's not autocorrelated, it looks very erratic and changing very rapidly. This one looks like it's changing more smoothly, and you have a series of increases and then decreases. But they have the same mean and variance apart from that. It's the same thing. So some people talk about the color of noise. It's a different, you have spectral analysis of these patterns of fluctuation, but anyway. So if you think of a mutation now, again, that has an effect alpha in the background phenotype. So M is the background phenotype in which it occurs. Again, defining things in terms of this logit. For Latinist is logit, I guess. So log of p over 1 minus p, the change of that will just depend, as I said, on this thing that depends on the effect of mutation and deviation from the optimum. And what's convenient of writing things in that way, and this was used a lot, especially by Gillespie. He studied a lot these fluctuation selection models when he was arguing that there's randomness in selection too. So it's also a stochastic factor in evolution. It's not just drift. But for what interests us here, it just ends up that you can sum all this past deviation from the optimum here. And so, especially if the optimum is following this Gaussian process, that's one of the main categories that you use for a continuous stochastic process. Then so does this logit frequency, just because of it's a sum, it's a linear combination of Gaussians, right? And also, if M doesn't change, the background doesn't change, then psi, this simply integrates all the past optima. It's just the sum of all these past optima. It puts equal weight on all optima. Okay, so with this, let's assume that the optimum follows a stationary, is the simplest version of something that's autocorrelated. It's like you draw the next environment from the same normal distribution as the previous one, but you condition on the previous one. So you say, I don't know, the environment at time t plus 1 you will say t plus 1 equals rho times the environment at time t plus square root 1 minus rho squared some x t plus 1. And this is drawn from a normal distribution with mean 0 and variance this. If you do that, you'll see that epsilon in the long term, it will have this variance, mean 0, and then the correlation between subsequent time step is given by this. Because this is an independently drawn thing. So this is a very simple way to model autocorrelation. If you do that, you can, so I didn't show how it's derived, but basically, okay, how to say that? Okay, the mean selection coefficient in this case it's the mean of this sum here. It doesn't depend on the variance actually. So if you change the pattern of fluctuations, so this expected trajectory on the logit scale is this blue thing. It's not changed here between here and here. It's not affected by how it fluctuates. It just depends on the mean optimum. However, the variance among, you could say the variance among replicates, then you end up having to calculate the variance of this sum. The variance of the sum is the sum of the variance plus all these covariances. But all these covariances, turns out, they will depend on these different powers of rho that just simplify and just have a, so it's just, you end up having some geometric series, sum of square at different powers. And so you have this very simple formula that eventually this variance of this thing depends on the variance of selection coefficient. This is regardless of whether it's with an optimum. It would work in other modes. The variance of the selection coefficient times this ratio of 1 plus rho over minus rho t. So the rate of, this is the rate of increase in variance among replicates, you see? So this says that when you have larger autocorrelation, you have more variance in this outcomes of selection. Yes?
Here in a mutation limited regime, or are there many?
On one locus, without considering the rest.
Because you had that background was MI in your equation in the previous one. So
I'll move on to the next. Yeah.
Okay. Thank you.
You can ask about this, but probably you won't be satisfied. Here the variance increases linearly. It increases linearly on the logit scale. As someone was mentioning earlier, I think it was Mike, when you place that in the actual frequency scale, this variance here means different timing of the selective sweeps. I think it's Nick. When you're crossing close to, when the logit crosses 0 is when you cross a frequency of around half. This is the region where you see selective sweeps, right? So this timing here, when you cross logit 0 is log, this equals 0 when p over 1 minus p equals 1. So when they're both half, you see? So what this translates into here is basically that you have very different timings of sweep because it spreads out. And it's really like more autocorrelation means you have a series of bad years or a series of good years. So this spreads the, when you sum up the selection coefficient sort of integrate and sum up this effect, then it causes differentiation on the trajectories. Okay, what I did also in that model is to use this kind of strange model where you assume that there's a background genetic variance for the trait, but you don't really model it. You just say there's normal distribution for that, and then mutation, it's causes a switch in the mean background phenotype. So you could consider this as a major effect in the middle of polygenic things. But if you're willing to believe this model, which makes some assumptions about independence between your main effect and the background, you get this strange result that actually here, this variance of this logit frequency, now it's become stationary. Instead of having the variance that keeps increasing, the variance, so in this case, the average trajectory goes down. And you see that the variance here, the outcomes, they remain bounded around you have this traveling wave somehow. So this is what it's shown here. Without background genetic variance, the variance of this, the stochastic variance of these frequencies, it's increasing over time indefinitely, whereas in this other scenario, it's sort of buffers, and you can have an analytical prediction for where it does. So do other loci, they buffer the stochasticity, persist? I don't know. What's happening in the model, what's happening is that the mean phenotype in the background is tracking this fluctuation in the optimum, and this actually this reduces the fluctuations of the optimum that's perceived by the mutation because it's sitting in the background that's already tracking the fluctuation in the optimum. It leads to that. So if someone wants to pursue this in a more explicit gen, I'd be curious to see what this does. I don't have much time, so I don't know. I just want to say a few things about this. You can stop me if you get bored or what. But so now, if you think of a polygenic trait that's responding to selection, this constant variance as before, the mean phenotype in the long run It depends on deviation from the optimum, etc. And you could show that in the long run, it depends on these initial conditions. This effect of the initial conditions will eventually vanish as time increases. And then you're just left with this sum of, it's kind of, this sum is the weighted average of all past optima. So the current mean phenotype depends on all the past optima, but with more weight on the more recent one. And then as time increases, the older ones, they get erased, if you want. The effect of the old optima on the current phenotype get erased as all the faster as genetic potential is high here. So in one way, so here, this is like fluctuations in optimum. This is actually for multiple traits, correlated traits, but it works for one trait. You think the green line is the optimum for one trait, the blue one is for the other trait, and this is what it does for the mean phenotype. It looks the same, but it's smoothed. This is actually, it's really something like smoothing a signal, actually. It's almost literally what's done when you smooth a signal, I think. So this is what I wanted to bring in yesterday about selection being some kind of signal processing in terms of, it's not exactly the same as storage of information, but it does somewhat process the environmental signal. Anyway, in more or less precise ways. So from this, you also get that you have this sum of this optima, and if the optimum it has a Gaussian process, then the mean phenotype also has this Gaussian process. So we're on this Gaussian world that we like, because it's a linear combination of Gaussians. So is the mismatch with optimum. So then in the end, the distribution of this maladaptation with respect to the optimum, when it's Gaussian, it can just be described in terms of this mean and variance. So that's good for us. And so when you have this fluctuating optimum at equilibrium, the mean phenotype will, the expected phenotype will be at the expected optimum. I can just wave my arms about it. This is the optimum, this is the mean phenotype. Overall, it's staying at the mean optimum. But what you're interested in is, if you take snapshots, how much it deviates from the optimum on average. This is the variance of these deviations that gets that. Also the autocorrelation. So one example to study that is, for instance, directional selection on the trait. This beta that I told you about. It depends on deviation from the optimum. Let's say the optimum is constant. Just because of drift, the mean phenotype will deviate from the optimum. And then selection will bring it back, and it will have this, as long as the population size is not infinite, it will not be at the optimum. It will fluctuate around the optimum just because of drift. Not too far, because the strength of selection determines how far it can go. Just because of that, you will have a fluctuation of selection gradients. Actually, if you measure selection, it depends on the direction of the optimum. It will depend. If you drift a lot, then you can end up very far from the optimum, and then you have selection to bring you back. So you can derive what would be the variance of selection gradients in this case. So this is kind of the lower bound for if you're measuring directional selection, how it fluctuates. Cannot be lower than this, because this is what drift imposes, that you're not at the optimum. So I almost wrote, this is a drift barrier for how, then I removed it because it didn't make sense. But this is the lowest fluctuation of selection you can get is the one that's caused by fluctuations around the optimum due to drift. And then the autocorrelation of this process is given by this function. So this is an autocorrelation that declines with time exponentially or geometrically. And basically, the autocorrelation declines faster when you have more evolutionary potential. So this is some kind of, this determines the evolutionary inertia, if you want, how likely it is that two phenotypes are going to be similar a long time, how rapidly you sort of erase the evolutionary history. Okay. When you have a fluctuating optimum, I think this might be a bit too much to take for people, perhaps, at this point, but you can also compute what would be the variance of selection gradients in this case, right? It's the variance of, it depends on the variance of deviation from the optimum. And so in this case, this shows that when there's higher autocorrelation in the optimum, it leads to better adaptive tracking, and then you have fewer fluctuations in deviation from the optimum and selection gradients. And again, drift will shift up or down these fluctuations. So skip. All I can say is that once, in some cases where every once in a while you have to check if these assumptions work because you know they're not supposed to work since you don't have a normal distribution with constant variance. Some people have shown how well it should work when it's not true. But in that paper 10 years ago, we did some simulations where we vary individual-based simulations. So this was actually some pre-SLIM simulation, since this person ended up working on SLIM. But this was his program that combined with the one by Philippe Messer. So he went across all kinds of different parameter values, etc. So basically, that's the thing, is that the expected variance that were, for instance, the variance of selection gradient in the long run, when you have, it tends to what you would predict in the mole, assuming constant variance if you use the right expected variance. Same for autocorrelation, etc. So at least in the situation where there's sufficient mutation, it works well. I think it works also. Sufficient mutation doesn't mean that you have to be in the Gaussian regime at each locus that Nick Barton was telling you about. It can be that you have many loci that are contributing to the trait. So I wanted to tell you about population size, but I think it's too late. But the only thing I can say about this is that these models with optimum, they also predict what would be the distribution of this population size when you have fluctuations. If you have many replicates, then since I just show this I think is important. If you look at the log population size, it will depend, the log population size at time t+1 in discrete time model depends on your log fitness. It's really just like n at t+1 equals w bar nt. That's what it's based on, when your w is an actual demographic thing. And so you take the log of that. So log at nt+1 depends on log mean fitness. And then selection depends on log fitness. So this is what bridges ecology and evolution. When you have the Gaussian fitness peak, then growth rate depends on the square deviation from the optimum. So this is a bit like what we had in Fisher's geometrical model. And this is what it leads to the same cause leads to the same result. So you have it leads to this gamma distributed population size, etc. I won't say more about that, but we've tested that with experiments. But this is not really about evolution, it's more demographic. So yeah, my conclusion about this is that these models of adaptation to an optimum phenotype, they rely on plausible biological explanation. They don't have to be necessarily exactly true, but at least they can predict adaptation across a range of conditions. This is also going back to my previous lecture. You're trying to reconcile what you observed for fitness effect of single mutations to what you observe for evolution of traits. You can combine several lines of evidence. And it can help us understand what happens in adaptation, of course, in natural settings where there's changing environment. To me, it's just a starting point because reality is more complex. We've seen examples where we had multiple peaks with this ecological speciation. You can see dependence. Space, I didn't even touch on. And then lots of traits respond to the environment, actually, as Luisa mentioned in her lecture. And so I'm going to be talking about that on Friday. Okay, thank you.
I think
it's time for questions. But,
I think you skipped over a critical point. It was shown in your slides at the end there, but it struck me, and that is that the effect of one generation of maladaptation can persist for a long amount of time. And that strikes me kind of as the ecological equivalent of a harmonic mean. It's one way to, I mean, not precisely, but the fact you can have a bad event that will persist for quite a while.
Thinking about this thing, this type of thing? This long-term, how it depends on the...
Yeah, it was in one of your summary slides that you went through. But
okay.
Right there. So yeah, past extreme events can have long-term consequences.
Yeah, it's just because it's summing.
No, I think it's a really interesting point.
I want to contrast, yeah, I just want to contrast what's happening here. Sorry. Here, the past events get erased because of this term. When you're going farther in the past, this is very small. So the past events get erased. And this is why you get this kind of smoothing here, because you're just mostly you're summing this. This is what determines how much it's smoothed, actually. Whereas for population size, it's not the case. You sum all these things with equal weight. It's actually more complicated when there's density dependence. You can show that density dependence does the same. When there's very strong density dependence, basically, you're really stuck at the carrying capacity, and the stronger density dependence is, the more you forget about past maladaptation, because now you're mostly, population size is mostly determined by density dependence. But I guess this is not the topic here. Well, you can ask me questions afterwards also, I don't know. My previous lecture was too short, so this time I thought I could talk about many things. So lunch is upstairs. Okay. Other questions?